And thank you for the organizers to inviting me. This conference was really great for me because I like conferences where I know only partly what's going on. So I think I know enough that I understand most things, but what I like is that I learn a lot. And I already brought a lot of ideas I think that I can bring into my lab. So I'm hoping to tell you an interesting story. Now, Ethan was telling you about that the university where he works sent him a letter saying that he either works on COVID or go home. I got the same letter, or a similar letter, and I decided to stay home. I became a theoretician. I went to the dark side, and so my talk will be mostly theoretical. And it was not a difficult transition because I have been doing theory all along, and I was always interested in theory. I always thought that I was bad at theory. And as you get older, I think you get more patient and you get better at this. Actually, at failure, you get better at failure. What I want to tell you is about how to count molecules in single cells. And it's actually a complementary talk to Johannes Holbein, right? Holbein. But I think it's more focused on eukaryotic cells, like counting molecules in eukaryotic cells. So as a motivation, the human genome has about 20 to 25,000 genes, which make about 60 to 100,000 different proteins. Now, this is an interesting number because it says about, right? 20 to 25,000. You would expect that from the Human Genome Project, people would actually know how many genes there are. It is actually not known, interestingly enough. I read just the recent literature, and I was actually at NIH doing the Human Genome Project. And it was funny because while this discussion was going on, I had some friends that worked in Celera, which was the private company that competed against the government. And they actually literally sold 60,000 genes to the industry, like the intellectual property. And then they found out that there were only 20,000 genes. So it is still not clear how many genes we actually have. 60 to 100,000 different proteins comes from splicing, right? So in the eukaryotic genome, you have patches of coding sequences that can be combined in Lego-like fashion. And that's why we have many more different proteins. But every cell expresses only a subset of these. So it's not like every cell has 100,000 different proteins. I just looked this up and I'm not sure exactly, this is probably also full of uncertainty. There's about 1,500 transcription factors, 10,000 gene promoter sites. But this can also depend on the cell line. I think overall, maybe this is the wrong number. But what I want to tell you from this is that a cell is a huge control circuit, right? I mean, an enormous, complicated control circuit that controls pretty much everything, right? It controls membrane potential, controls lipid composition of the bilayer, the lipid asymmetry of the bilayer, everything. So, and we know this from the engineering. I'm in an engineering department now as a physicist, but still I'm an engineer. And as engineers, we think about, you have a system, you don't understand this is a black box. You want to figure out how this works. The way you do this is you have to understand the dynamics of a system, right? You have to follow the dynamics. You have to get time series of stuff, right? You have to measure as many components as you can, and then you take that and analyze it. And so what I'm saying essentially, I mean, the best thing would be to measure many protein concentrations as a function of time. Johannes Holbein already introduced GFP. There's some other labels that you can use for RNA but GFP is a great tool, right? Because you can fuse it with pretty much any protein, and then you see fluorescence. So there are a few problems that have already been solved, I think. Actually, Joachim over lunch told me that he can do it even better now. So Joachim over the years has been developing these pattern surfaces, and these pattern surfaces. They're made essentially from PEG, right? So the gray is, the dark gray is pegylated. cells don't like to adhere on peg. And then he has these fibronectin-coated squares. There's 30 microns here, but it depends on the cell, how big the cell spreads or something like this. But the cells really, because they don't like peg, they just crawl to these spots and then adhere, right? And then spread out. And the other advantage of these patches is that these are squares, they have corners, and then the cytoskeleton, locks into these corners and they don't rotate. And the big advantage from an analytical perspective or quantitative perspective is that you don't have to worry really about the shape of the cell changing over time, right? It's sitting on one place. You can take a few voxel pixels around it, but you don't have to follow a cell. And if you actually look at work where people follow cells and then look at fluorescence, it's very, very noisy. And that's sort of solved by this technology. So this is one trace of a GFP over time. This is actually work I think I was on my sabbatical, but Joachim, I helped him a little bit with the data analysis. So the idea of the system is very simple. So you have a cell, you transfect the cell with messenger RNA, very similar to the COVID vaccine. You throw a certain amount of messenger RNA in here. The messenger RNA codes for GFP. There's a translation rate from messenger RNA to GFP. GFP initially is not fluorescent, so there's another rate constant that transfers it from non-fluorescent to fluorescent. So this is maturation. And then what we put in is decay rate for mRNA, decay rate for protein, and we assume that the fluorescent and non-fluorescent is the same. Right? This is the simple model. This is a differential equation that you can solve. And you can fit the curve here. Right? So this is each individual cell is being fit, right? And then what you get out are the constants of messenger RNA decay, translation rate, protein decay, and maturation rate. Now, what's interesting is that, two things I want to point out. Two things I want to point out that the fit is not perfect, right? The fit looks good, right? For most people, the fit would be sufficient. But as a data analyst or theorist, I would say this is a really bad fit because it goes under and over, and that's not how it should be. The next thing, what is interesting is if you look at the distribution over many cells, you see that the rate constants that we get out of this model varies quite a bit. If you look here, this is almost 100% width of the decay rate of the protein. This is the maturation rate is about 50% error. So there's a lot of variation, right? And we know this. Biology knows this for a long time, that cell variability is a very big and important thing, actually. And so one thing that I want to motivate you with, because this is something that I'm working on, is can we describe this variability in the biochemical reactions? So one thing that we all know is that cells regulate things. This is homeostatic regulation. So the cells set a certain concentration, a certain rate at a certain level, and then the fluctuations around this level, but it should always come back to that homeostatic point. And so what you can do is you can just say, okay, instead of fitting a single translation rate, we just make this is actually called an Ornstein-Uhlenbeck process. This is essentially a Brownian particle in a harmonic potential. And you can make this fluctuate, and there's two parameters of that. It's the variance of the process and the relaxation rate. Right? So the autocorrelation function of Ornstein-Uhlenbeck is an exponential. And then what you can do is you can take this and then use it to simulate your protein expression, right? And every single time you do this, there's a different process, right? Because it's a random process. And you can see that with the same parameters, essentially, I can explain this curve and it can explain this curve, right? And then this is actually the ensemble. If I run this like 10,000 times, then you can measure the ensemble. And that's what we want to do, right? We want to take the data and then figure out whether from the data we can extract the mean of these values, right? And also whether each individual cell accesses all these translation rates, right? Because it's It's an average over the ensemble, but the question is, this is always a statistical physics problem, right? The time average versus the ensemble average, whether they're the same. And in cells, they may not be the same. So that's something you can address with these kinds of analysis. I also wanted to show you something interesting. So I got this data a few days ago. So I didn't analyze it and I can't show you anything about them. But this is actually real deep work on understanding cells. So this is work by Gabor Balazi. He's also at the Laufer Center. He's a Laufer professor or something like this. So he's half-time at the Laufer Center, half-time at biomedical engineering. And then this is Rafal and Jonathan. Rafal was actually postdoc with Joachim, so he came to Stony Brook. And he did two experiments. So one experiment was there is a gene that expresses GFP. You can turn the gene expression on, and you get essentially gene expression. And the gene expression is given essentially by so how much protein you produce and how much protein gets decays, right? And then you get essentially a steady state. But you see that there's a lot of fluctuations, right? And if you do the autocorrelation function, right, you see that it decays about in maybe 10 hours or something like this. And this looks very much like an exponential. Then he tried something else. So this is a system that is actually a bacterial system that was put into a cancer cell. But this has a negative feedback loop. So the trick is that you have a promoter that then produces inhibits the translate which I'm stunned here. So it inhibits the production of this promoter, right? So it's a negative feedback loop essentially, and this is often used in biology to control concentration, right? So you get a concentration that is much more stable. And you can see this, but the other thing that you see here from the autocorrelation function is that it has a dip. It is a real dip here. So that means that actually, if you have an increase in concentration at time 0, you expect to have a lower concentration, right? So you have a fluctuation that goes up, right, in concentration. After a few hours, right, after about 20, 30 hours, you're expecting to more likely have a lower concentration. And we think that comes really from the fact that this feedback loop, right, you produce essentially a protein that then has to interact with DNA, right? And has to actually come into the nucleus and diffuse into the nucleus. So this is just a motivation. Okay, so I want to spend a little bit of time on how to do data fitting. I told you that I have become a theoretician, and actually I have become a computational neuroscientist. But fortunately for you, actually, most of the data analysis methods that we use for computational neuroscience is also applicable to systems biology and quantitative biology. So what I want to tell you about is Bayesian parameter fitting of time series data. The assumption that we always make is that all the time series time points are Markovian, so that x2 only depends on x1, but not on any previous points. In general, when you have data, if you have a model, or if you have what we may call the truth, then this can typically be described as a stochastic differential equation, right? So there is a derivative that depends on some function of x and t and some parameters. And that's a deterministic part of the stochastic differential equation. And then you have another term that is the stochastic part, right? And then you add essentially a random walk to that. There's a linear process. And then you essentially create this chain, right? You say, okay, so I have x1, and then I have x2 and x3, to xn. But what I really measure is actually something different. I measure x1, but with noise, and actually possibly with some transformations and things like this. So what we typically say that y is a normal distribution that has a mean around this real value with a sigma, right? And this is a typical time series. So the goal is to actually get the posterior probability of the parameters given the measured data, right? Which is P of the parameters given the data. And I put the sigma here because we often know the noise. So if you know the noise, you can put this on the other side. And here are a few thoughts on this. So the posterior probability, in order to get the posterior probability, you essentially use Bayes' theorem. You can turn around the conditional probabilities. You can flip this and this, right? This is done by Bayes' theorem. Now imagine this turns out to be very hard to do. Joint probabilities are easy to calculate, right? Because P of X, if you know X and Y and lambda, then you can calculate the probability, right? This comes back to this, right? So if I know X1, all the X's and all the Y's, I can calculate the probability that this ever happened, right? So that's easy to calculate. Now what we want is this, right? The probability of what we measure, or yeah, this is the likelihood, or we want actually the opposite, but that's easy to turn around. So that's hard to do because you have to integrate out all the X's, right? And if you think about it, so if every data point that you measure, let's say you have 200 data points, this is a 200-dimensional integral, right? It's very, very hard to do. But what comes to the rescue is actually modern statistical programming. And I want to actually run a little advertisement here. If you have not seen it, then you should really look into it. Julia language. So Julia language was developed like 10 years ago by MIT. It, I think, is viable, has become viable about 1.5 years ago. So we actually started when when 1.0 came out or something like that. We are now at 1.8. The idea of Julia is really that it is a language for scientific computing, high performance computing. The motto is walks like Python, runs like C. It's a just-in-time compiled language, runs essentially like C, sometimes actually runs faster. It's open source, right? MATLAB is not. MATLAB costs money. It's actually better than MATLAB in many respects. It fully supports automatic forward and backward differentiation. This is something that is very important in data analysis nowadays, right? Any machine learning procedure, right, uses automatic differentiation. Have you ever asked yourself why TensorFlow is called TensorFlow? It should be called machine learning, right? I mean, machine learning.py or something. TensorFlow is actually a wrapper around automatic differentiation, right? TensorFlow. Flow is derivative of something, of tensors, right? Because Julia is constructed in such a way that any function written in Julia is automatically auto-differentiable. So you don't have to worry about anything, right? It automatically compiles the code and calculates automatically the derivative of anything. Differential equations.jl is a package that has absolute top-of-the-art solvers. I think it's the best out there right now. You can look at the benchmarks, you can look at the accuracies. It's really amazing. They have solvers for differential equations, for stiff, for non-stiff, for jump differential equations, delay differential equations. Pretty much anything that you want to do. And it's incredibly fast. Just anecdotally, my student came to me a few weeks ago. He said, oh, he got some MATLAB package from some collaborator on some neural mass model. This was like 200 differential equations, coupled differential equations, nonlinear coupled differential equations. He wanted to solve it. He wanted to solve 10 minutes of data. It took him an hour, right? MATLAB. So I implemented this. It took me about 2 hours to implement it. It ran in Julia in 21 seconds with a better solver. I mean, this is the amount of time that you can potentially save. It has to do with that MATLAB is also a scripting language, right? So loops are very slow. Modeling toolkit I use a lot. It's a completely symbolic math part. So you can write all the differential equations, everything you can write in symbolic form. It can simplify your equations. It can, find all the states. It also can calculate Jacobians, right? So if it's sparse, then it calculates the Jacobians for you. you can run it much faster. Sometimes you get, you know, a 100-fold increase in speed just by doing it symbolically. And Turing is the probabilistic modeling and sampling package. Okay. So now I wanted to show you just an example. This is what I would call extreme fitting. This is a typical model for modeling neural data dynamics. This is actually a simple Van der Pol oscillator, you know, from 1920. Simple differential equation. I added some noise terms here. So you have essentially a noisy Van der Pol oscillation. Then I add some noise to that. And then to make it more difficult, I just apply a sigmoidal function to that thing, right? So everything becomes highly nonlinear, right? So this is the sigmoid applied to the data that you saw before. And then we run a data fitting on this. So this is used to automatic differentiation variational inference. It takes about 2 minutes. If we do Monte Carlo, it takes about 4 hours to run because it has like 400 parameters or something like this. But this is how fast you can go. So there's no excuse anymore to not use Bayesian analysis methods if you want to do a good job in fitting the data and getting actually the proper confidence intervals for your parameters. So it has gotten really fast. Okay, I want to get back to what we want to talk about before. So I want to ask what technologies, what advances are necessary to study complex biological circuits, right? So we want to make as many colors as possible. I think we saw already in talk Johannes Holbein said, you know, I mean, I think actually more realistically, maybe 3 colors, actually almost only 2 colors, because cells really don't like light. So longer wavelengths are less harmful. One could possibly distinguish brightnesses in lifetime. Lifetime, the only problem is that you need really complicated equipment. You need femtosecond lasers. But brightness would be a nice thing. So if you could have, you know, 2 or 3 colors with 3 different brightnesses, then you would be much more able to multiplex. The other problem is accurate measurements of numbers, right? Total fluorescence is the number of molecules times the brightness. Now, if you don't know the brightness, then you're in trouble. And it turns out that the brightness is actually depending on the environment. So a lot of times you have to actually get the brightness in the environment that you're measuring in. So so for example, if Joachim is calibrating, you know, with a microfluidic channel, you don't necessarily exactly know how many. The other thing that I want to point out, and this is sort of the only the only place that has nanometers in this, because what I'm going to tell you is independent of the length scale. It is generally true. So let's think about single molecule detection in general, the choice of detection volume, right? So we know Avogadro's number. So what we can do is we can calculate what is the volume, right, at which what is the concentration at which a particular volume, right, has 1 molecule in it, right? So 10 micrometers cubed is a picoliter, right? At 1.7 picomolar, right? This is sort of because the arbitrary, you know, choice of the Avogadro's number. At 1.7 picomolar, you have 1 molecule per 10 microns, right? Femtoliter, you have a nanomolar, right? So this is typically FCS range. So if you think about the focus, attoliter is micromolar and zeptoliter is millimolar. So if you think about it, if you want to really calculate or measure concentrations, you have to be in the right range, right? And if you think about it, I mean, millimolar, I think this is 1 molecule per cell, right? Approximately. This would be 1,000 molecules per cell. This would be 1,000,000 molecules per cell. So we are sort of in this range, right? So we definitely have to confine our detection volume to 100 nanometers or something like this. Okay, now I'm coming to the sort of the heart of the talk. So how to measure concentrations? So the best way of measuring concentrations, if you think about it, would be if you had a box that is illuminated, right? So you have some intensity here, and only molecules that are in the box are being counted, right? If you could do this, then you would be golden, because all I have to do is to do a histogram of intensities. And then, for example, if on average there's one molecule in the box, right? I set the concentration so that there's one molecule in the box. Poisson tells us that approximately the probability of finding 0 molecules is 0.3, something like this, a third 1, and a third more than 1, right? And just by looking at the distribution, I can actually tell you what is the concentration of the molecules in the box, right? But the problem is that we can't do a box, right? It's very, very hard to do a box. So what you typically have is an exponential, a Gaussian illumination profile, right? And I wanted to ask, what is the intensity distribution function over a Gaussian, right? And I thought this has been worked out or something like this, but it hasn't. And I want to show you the results of that calculation. And I actually had a long discussion with myself whether I should show the results first and then show you the theory, or first do the theory, but I think I show you the results first. And then I hope you're intrigued enough that you will follow along with the theory. So these are the simulations in 3D. So all I did is I made a big box, I put lots of particles in there, I let them diffuse, I put a little Gaussian in the middle, and so on. And by the way, I have to say that the 3D case has been treated in the literature. So there's papers from 1999. There's actually 2 groups that simultaneously did these kinds of things. And they were asking exactly the same question. They were saying, can we, by looking at the shape of the photon count, it's called photon count histogram method, can we look at the shape and figure out how many molecules are in there? And you can see there's some differences, right, between concentration 0.2, 0.5. This is essentially the number of molecules per detection volume. You can see this is asymmetric, and if it gets too high, then it becomes a Gaussian, right? Nobody would argue about this, but you could also immediately see that these shapes are not very good, right? They're not very distinguishable, right? Whether this looks like this, I don't know. It's very hard to say. Now let's get to 2D, right? Dimensionality is very important. Go to 2D. Suddenly, something really weird happens. So there's at low concentration, you see this shoulder here. This is a place where you can take the derivative, right? It just falls off suddenly. And interesting at 1, right? So this is great because this can be used, right? So if I had a distribution function like this, I could look at the fall off, right? I have 2 molecules, they just add together, right? And then I can see 2 edges, and then I can tell there's 2 molecules in there, 2 different kinds of molecules with 2 different brightnesses. And I can tell each other's concentration, right? Because if I know the intensity, by the way, I assumed in the simulation that the amplitude is 1, right? Okay. And now I'm going to show you 1D, and this will really freak you out. So 1D, that's a simulation. Looks like this. So it has a divergence at 1. So it's more likely, right, to have intensity of 1, to be in the middle of the focus. And I said, I couldn't believe it. I still don't know why this happens, but it's true, right? You can see that this actually persists almost up to 2 molecules per focus. So there's these peaks in here. And I have to say, I didn't believe this, right? Because it looks so weird. And I hope I motivated you to actually look at the theory. So I have 5 more minutes. Okay, so let's go to the theory quickly. It's actually, you should be able to follow along. So the first thing that you can do is, this is a very hard problem without putting limits on it. So what I did first is I took a box, right? So I made a box. I cut off the Gaussian and I said, okay, I put 1 molecule in here and ask, what is the expectation value, right, of the powers of that intensity that I'm measuring? And it's super simple because, you know, it's just the integral over Gaussian. The edges here give the error function, right? And it's proportional to the power to the n, right? There's the square root of n comes in here because the square root of n comes in here because of the Gaussian. So you integrate a Gaussian, the prefactor essentially will be a square root of the coefficients of the exponent. Yeah, because if you take the power of this here, then the n comes in here, and that gets to a square root here, right? Okay. Then I have to ask myself, so how do I treat two molecules, right? So how, you know, one molecule is easy, right? How do I do two molecules? So now you have to look at probability theory, right? So if you want to know the probability of a intensity that consists of two intensities, right? Then this is just a convolution of the probability that rho A is X and rho this is a phi, right? Phi B is, I didn't take Greek. Phi B is phi minus X, right? So that the sum is always phi, right? So in math, you probably learned if you have a convolution, it's much easier to do this in Fourier space, right? So what we do is we go use the characteristic function. So the characteristic function is just the Fourier transform of the probability distribution, right? And it turns out if you expand the exponent, right, you can actually see that this is just, if you do the series expansion of 1 plus ik over 1, the expectation value of phi, right, and phi squared and phi. So if you know all the moments of a function of a probability distribution, then you can calculate the characteristic function, and then you do the inverse Fourier transformation of the characteristic function, then you get the original probability function out. By the way, this is not always true, right? There are actually different probability distribution with the same moments that are different. So you have to be careful, but in this case, it works. And then you just put everything together. So the characteristic function is 1 plus this here, right? Because I can just add up all the terms of the expectation value of the moments. Okay. Now this looks very, very close. Look at this. This looks very close to an exponential function, right? So this almost looks like an exponential function except for the square root of n, right? So we expect it to sort of like oscillate or something like this. The next thing that I want to do is I want to take the limits, right? So we had this box that was closed. Now what we want to do is to express it as a function of concentration, so n over L. Then we can take the limit. We can take the limit of the error function going for n is to infinity goes to 1. The limit of n to infinity, 1 plus x over n up to n is exponential. So we can take the limit of this characteristic function to the power of n, which means n particles, right? So, and that's this function, right? So you have a simple function that looks like this. Everybody get? Okay, now let's look at the dimensionality. So this is 1D, right? And all that happens for 2D is there's another square root of n coming down here. So you have n factorial times n, and in 3D you have n factorial times n to the 3/2. And the prefactors, right, because of the integrals there's some prefactor changes. Okay. A side note, quick side note. It turns out that for 2D, this is actually connected to a special function, which is called the exponential integral. So this is e to the minus t over t times dt from z to infinity. And you can also take the sine integrals, the cosine integrals. You can actually see that this is exactly the same form, right? Factorial times n, n factorial times n, the same over here. There's this funny constant in here. It goes like gamma times ln k. This is the Euler, actually this is called the Euler constant, right? It's not E. E is not the Euler constant, but this is the Euler constant. But unfortunately, all these special functions are very, very hard. I mean, I don't know whether you know this, but these series expansions are very hard to calculate, right? Think about sine. Let's say you want to calculate sine at a very large number. What is going to happen in infinite series is that you have numbers that a large number minus a large number plus a large number minus a large number and stuff like this. This can go on forever, right? Until it decays off. So this is actually not a good way of calculating these functions. And that's why I had to go to my friends, to my mathematician friends, and they immediately saw that you can express square root of n as an integral. As it turns out that 1 over square root of n is the integral exponential da da da. And then you can turn this whole thing into an integral, and then this becomes essentially the characteristic function. These are high energy physicists, they're useful for something. And then you can essentially, make it a little simpler. But here's the result. So this is the simulation theory, rho is equal 1 for 1D, shows the same peak. By the way, I have better pictures. The sharpness of the peak really depends on how far in k-space you go. But if you sample more k-space, then you get sharper peaks. We can make this as sharp as we want. So it's perfectly in agreement with the theory. We can actually predict all of the shapes, right? And exactly at the right concentrations, right? They're completely overlapping theory and simulation. Same for 2D. Same, right? And I think I'm probably out of time, but I just wanted to address the following questions of how, actually, maybe I should spend a second on this question, right? So you should ask yourself, how would I do this experiment? Right? So we have a focus in 3D, right? But if you actually look at the theory, a light sheet would work. A light sheet is essentially a one-dimensional Gaussian, right? Where the other dimension is constant, right? That's perfectly valid. So if we have a droplet or a cell, we put a light sheet through it, right? And then do intensity measurements, that would work. The 2D case, I would imagine you could make essentially a very long focus, right? Like very similar to light sheet, you could make a very long F lens, right? You could make a very long focus, right? And then use a small part of that to do these measurements in 2D if you wanted to test the theory. This is sort of one remark how you actually measure intensities. Typically, when we measure intensities, these kinds of experiments, you use a single photon counters. We all know that actually what you see is actually not single photons, but it's just an exponential probability of intensity. So something like this. So you have to convolute this essentially with your probability distribution function. So the fitting would be essentially taking in raw data like this and then calculating the posterior probabilities of all these parameters of the distribution function. Conclusion: use Julia. My first advice. I think intensity distribution function can clearly behave differently in different dimensions, so we should exploit this. I think 1D and 2D really can use to calibrate fluorescence measurements. So if Joachim wants to know, how bright your GFP is, this could be a nice method if it works. And we should be able to distinguish between different fluorophores and different brightnesses. I want to give my collaborators. So I was actually paid by CeNS, so I want to acknowledge that. So Joachim Redler, Daniel, and Anita, and Rafal were working on the data fitting, and Stony Brook University, Gabor Balazi, Joshua Azuka, and Lilian Mojica Porodi. She's my wife, and she's also my boss now, because she wrote all the grants for the computational neuroscience. And then we have a collaboration with MIT, with Alan Edelman, who is the inventor of Julia. and Chris Rackauckus, who is probably the world's expert in solving differential equation, and he's the driving force behind the success of Julia. And I'm open for questions.
Thank you. I would left us with many questions.
Okay.
Questions, please, from the audience. Maybe, yeah, Johannes.
Perhaps for applicability in a real world setting, how would background counts and or autofluorescence of the cell affect the method?
I mean, but you can put this into your model, right? I mean, when you model this thing, I mean, a constant background, I think, is no problem whatsoever, right? Because you can just put that, you know, as a noise. You can do something very similar to what I showed before, right? So you can just say any data point is not exactly what it should be.
If you have a model like for Poissonian, I get it, but for
Yeah.
autofluorescence of a cell, is there something known, a model for the statistics of the photon counts that come from the background?
I don't know, but I would just actually, you know, take a cell without fluorescence and look at the data and model it right there. I mean, this is pretty simple, right? So you can just look. I mean, so the question really is whether there is a lot of. I mean, what you want to look at is fluctuations. What is the photon counting histogram of autofluorescence? And I mean, I would just assume that it's just a constant intensity. But I don't know. I mean, there's also bleaching, right? I mean, the question is whether this bleaches. But this can be very easily put into these models. I mean, this is not especially with the powerful nature of the probabilistic programming that we can do now. I mean, the field, they're fitting, you know, up to 1 million parameters. No problems asked if you have enough data.
Yeah. Maybe. Yeah.
When I was a graduate student, if we had more than 3 parameters, there was no way.
Okay. Yeah, so could you maybe go back to a slide at the beginning where you show a simulation of a fluorescent readout over time?
Oh. Fluorescent readout over time. I don't know what you're referring to.
It's right at the beginning, I think. There, and you just press it.
Here?
No, forward.
Oh, here.
Here. So you said you were introducing noise. So my question is rather, did you perform a lot of simulations and for each one a new noise term, or is the noise during the time course?
Yeah, so that's really the question, right? So the question is, why does this curve deviate from the mean, right? So this is a curve, right? And this is the best representation of a differential equation that we can find, right? With least squares, right? Mm-hmm. But you can see that there's a clear deviation. So what changed, right? So some parameter must have changed in time.
Yeah.
Right. So for example, if the translation rate changes, right, you could expect that maybe something like this happens, right? And so what I was trying to do is to say, because we see a large distribution in cell population space, right? You could either say that a lot of people, they have different properties, you know, they have different translation rates, but it could also be that every cell fluctuates in translation rate. And so what the model that I suggested is a fluctuation, let's say, in translation rate. So what you would need to do is to make lots of models, right? So you make a model for translation rate, you make a model for decay rate, and so on. And then you look at which one is the best, right? And the nice thing about Bayesian analysis is that you can always tell which model performs better than another one.
Yeah.
Right.
So yeah, you answered my question.
Okay.
other questions? Maybe I can ask one question because you mentioned Julia and the power of Julia and also TensorFlow in this context. So I wonder, nowadays people use GPUs in order to increase computer power for artificial intelligence. So how does it compare to using Julia?
Oh, I should have told you. So this is one thing that I didn't mention, but this is actually probably the most powerful feature of Julia. Julia was designed from the beginning to be agnostic to any computational resource. You can just tell Julia, I have so and so many GPU cores, I have so and so many CPU cores, and it figures out what to use best. It's actually very efficient. So I would actually suggest to you, I mean, as an advice, I looked at the field a little bit. You should not buy any big cluster computers anymore. No, seriously. The cloud is so good. The cloud is so good, right? Because if you buy a big computer, it's going to be old in 3 years, right? Or in 2 years. Let Google and Amazon and stuff like this figure this out. Seriously. So now Julia actually has an interface. So it's called Julia Hub. You go to Julia Hub, you open an account, you put $20 in there or something like this, and then you can just use as many computational resources as you want, right? And it allocates whatever you want. Okay. And this is, I think, the model for the future. Academia is not yet, we can't really pay for this, unfortunately. So I had actually trouble, doing this with my grants. But this is the future. I mean, I see so many computers lying around in the labs and that are not used anymore because they're obsolete. Why waste these resources, right? If you have Amazon or whatever company this does,
Just realize we're running out of time.
Yes, we're running.
We need to close the session. I'd like to thank you again very much for the presentation.